Thank you, Michael. Thank you again, Ray. I plan to allow discussion and comment from the floor till around 2 o'clock, then allow each speaker 5 minutes to sum up, and then we'll make way for the next session in here, which starts at 2:15. Bob, do you have a question or a comment?
Yes, I have a question, actually, just briefly, on Michael's. I also put my cards on the table. I'm scientific director of Frame. I ain't got food. And I haven't asked Michael this question before in person. The question is very simple. Do you hold your views that animals are still required in experiments because you're a trustee of Frame and because Frame gets funding from industry, as is implied in this document?
Absolutely no. I don't get any funding from industry. I'm an independent person and I speak my own views, not the views of Frame. And of course not. It depends on the circumstances. For example, the example I gave of testing batches of insulin preparations for potency. That, in a sense, is validation by use. It goes on over many, many years. And therefore, I would say that is reasonable validation. There are other circumstances where maybe it's not. So I think each case has to be judged on its merit.
Could I ask a
Yes.
question? Would you support proper validation under the current standards that were arrived at in Solna and through exam and exam? Would you support those validation criteria for new animal tests or revised animal tests?
Again, it depends on circumstances. I mean, you look at the example I gave you of the liver tumours, where a microwave probe was used to develop a new treatment for liver tumours. In a sense, the validation of that was when it was first used in patients. So there's a very, very short period from the development of a technique to its actual use. But of course, in drug development, this takes years and years and years. So I don't think you can give a general answer to that.
You like to come and sit up here?
Can you give me an example of an animal experiment that you would not support simply on ethical grounds?
Well, anything that is trivial, anything that is badly designed. I mean, I am personally quite critical of much of biomedical research, and if you like to come to my symposium immediately after this, you will hear that I will give some critical comments. So anything that is not scientifically valid, and anything that obviously creates excessive amount of pain for a trivial gain, I would say I wouldn't support.
Any other comments or questions?
My name is Ken Shapiro. I'm a psychologist for the ethical treatment of animals. And in an earlier symposium, I made some comments about the use of animal models. And both of you have emphasized the issue of whether or not the model and the model are similar or different. And my thesis is that that kind of discussion, the degree to which the model and the model are similar and different, is not the bottom line problem. If validation simply means discovering similarities or confirming similarities, then that's not a very helpful bottom line criterion. The better criterion is, did you learn anything new? You can validate the stuff that you knew to be in the target and find it in the model or vice versa, and that's not having learned anything. You also can learn from differences. So we need other criteria besides validation. One of those criteria is, do we learn anything? The second is, do we gain in effectiveness for treatment? And there are simple ways of ascertaining these considerations. One is a citation analysis. Did the animal model literature receive citation levels comparable to other studies? did they receive citation levels in the relevant clinical specialties where they're supposed to be applied. We can also do surveys of the relevant clinical specialists. Do they know about these animal models? Do they find them useful? So it seems to me that this discussion of similarities and differences goes on forever. It's half empty, it's half full. Ray says they're different. You say they're similar. Back and forth, we go to different levels of analysis, biochemical and so forth and so on. That's not where it's at. Where it's at is, did we learn anything? And was it to regain in treatment effectiveness? And there are simple social, scientific, analytic tools to determine that. What we need is researchers to go out there and study particular animal models, find out the level of citation, interview the appropriate clinical people, and find out whether they learn from them. That will give us a good scientific argument about the science used here. That's, of course, bracketing ethics, which I don't do. I think it's unethical to do this research. That's not the discussion here.
Can I comment? I agree with you, Ken, and I think if you read one or both of the books, thank you. And you know that I quoted that study, that citation study that I think you're referring to. And I think it's in the second book in the neuropsych chapter. So I agree with you. But this, without getting into the old MD versus PhD argument, is basic research good in and of itself, or does it have to produce something? Okay, without going down that direction. What we tried to do in book 2 especially was do both things. Okay? Coming to you today as a clinician primarily and someone who has taught in 2 medical schools, I can tell you point blank a vast majority of my colleagues think the animal model is a waste of time. And this goes across party lines. It goes across all ethnic groups, male, female. I mean, it's just a joke. And if you really want to see some people get upset, talk to some physicians who have recently had relatives or a close patient die, and tell them how much money went for breast cancer or Alzheimer's, for the animal model. So I agree. I'm very happy to see empirical data. And indeed, a vast majority of Book 1 is empirical data. And furthermore, I would also say, you're shaking your head. You don't think a vast majority of Book 1 is empirical?
No, it isn't empirical data of the kind I'm talking about. It's simply descriptions that show differences. It doesn't say whether those similarities are differences.
Okay, that leads me to the second point. The second point is, why are you doing the research? Okay, the examples that we use and will continue to use are examples where the animal model failed. They're peer-reviewed references, et cetera, et cetera. Now, if you're doing animal experimentation in order to help human beings, then show me the data. Show me the data that says it helps. And I think that's where you and I are agreeing on this. I'm saying that when I quote studies out of the New England Journal and JAMA that show that based on an animal model of stroke, 38 people died in the experimental group and 14 died in the control group that didn't receive the treatment, that's as good as it gets. Now, if you as a psychologist want to then go into the kind of psychological, sociological domain and ask physicians, I agree. AFMA, even as we speak, is sponsoring a very large poll in the United States and is asking those exact same questions. But as a scientist, I don't really care what my clinicians say. I want to see the peer-reviewed data. And if 198 drugs are released and 102 are relabeled or recalled, that's significant. And I'll let Michael disagree.
Well, it rather depends what level we're talking at. I mean, we heard this morning about how many species have now been sequenced and how useful it was to, as it were, explore the genomes of different species and get new information from them. And these are basically, therefore, being used as models of humans. And I think that sort of exploratory research is excellent and very good. And I tried to make the point that quite a bit of animal research is exploratory in nature. We are looking at things like mutations in that obese mouse to try to find out what happened. Sometimes this will give us information which is extremely useful for humans. In the case of the obese mouse, it tells us that there is a compound, leptin, which is produced by the white adipose tissue, which controls appetite. And we can go on and discover other genes that control appetite, as is being done now. And maybe one day we will find a way of controlling human obesity. And this is the sort of research that goes on extensively, using animal models, using not only vertebrate models, of course, but lower animal models. People are not pumping lots and lots of money into research on the worm C. elegans because we want a better world for C. elegans. They're doing it because it will tell us a lot about humans. For example, in my institute, we're studying apoptosis. Apoptosis is programmed cell death. Cancer is a disease not only of multiplication of cells, but of failure of cells to die. We want to find out how we can cause cells to die. C. elegans maybe will help us, because a lot of the genes that are responsible for the cell death in C. elegans have been identified in humans. So that sort of model, I think, is extremely valuable.
Time for one more question.
I have one comment to make. I didn't hear anything about the validity that we distinguish in drug research. That's the construct validity and the place validity of things. And I think you are mixing these constantly. And then you can compare them. Construct validity means that you have the same biochemical basis. And place validity is that a disease looks the same in humans and in animals. You should put these two apart and not mix them up like you did with the airplanes and the engines.
I don't think I mixed them up. I think that complex adaptive systems have causal functional dissimilarities. And I think that in and of itself invalidates extrapolation. I also think that causal systemic disanalogies exist, which also invalidate animal models. And again, I think, and Michael can feel free to disagree, but when I participate in discussions like this, what I hear are two entirely different conversations. Okay? If you want to have a conversation about whether or not animal models are heuristic, that's fine. We can have that conversation. That's not what I'm having today. What I'm telling you is that they don't help me as a physician. Now, I'm telling you that animal models are not CAMs. They're not causal analogical models, which the animal experimentation community maintains that they are. Now, look, there's a lot of things that are debatable in this venue, but there are some things that are not debatable. And until both sides of this issue can come together and agree on the fundamentals, you're never going to make any real progress. And as long as people continue to confuse hams and cams, which is what you're accusing me of, by the way, no, it's not going to happen. But if you're talking about biomedical research on animals in the hopes of curing human disease, and my God, especially if you're talking about drug testing, that is the use of animals as cams. There's no way you can say that's heuristic or anything else. If it kills the mouse, it doesn't go to human trials. Okay? And that's what I'm telling you is doing far more harm to my patients than good. And if you disagree, show me the data. Sacred Cows has about 500-600 references, Specious Science plus or minus the same. I figure half of them are from peer-reviewed sources. So I think there is a huge amount of data supporting my position. And I would just note, I was going to save this for the last 5 minutes, but I would just note, in my presentation today, you saw reference after reference after reference. Nature Biotechnology, Nature Medicine. I think the oldest one was 1996. I didn't see any in Dr. Festing's.
You didn't see a reference to...
I couldn't see it.
There was a reference to Nature last week.
Last week. And how many references did you have in your presentation?
Oh, I don't know.
Okay. I couldn't see the screen that well. But my point remains, and I make the point also in the handout, if anyone cares to read it.
Ladies and gentlemen, we're about to enter the last phase of this point-counterpoint discussion. I'd like to invite each speaker in 5 minutes each to expand on what they said in their opening remarks and to comment and explain on anything that they've heard either in discussion or the opposing speakers say. And on this occasion, Michael Festing will speak first.
Okay, standing up here, I'm really not quite sure what I'm going to say. Ray's paper really didn't tell me anything at all. He uses quite a lot of long words like cams and hams. I think La Follette and Shanks introduced these terms They use quite a lot of what I call straw men. In other words, they say scientists believe such and such, and then they tear down what it is that scientists, they say, believe. But in fact, scientists don't believe those things. They are straw men. They're not what scientists do. Again, I think, again, with La Follette and Shanks in their book, they are two philosophers. They have used the terms of philosophy to investigate how biomedical research is done. Now, what is the raw material of philosophy? The raw material of philosophy is what people have said. So what they've done is to look to see what people have said, and they've taken selected quotes from wherever they wish and tried to build a philosophical argument. What they haven't done is to go and look at original publications. And I would suggest to some extent Ray Greek has not done that, because in his book a lot of the references are simply to anti-vivisectionist literature, and much of that is quite unreliable. So I ought to respond to one or two things that he said. Again, the 747 model is something that La Follette and Shanks have brought up. They say that models can't produce anything new. Because a model of a 747 is produced as a result of copying a 747, we can't learn anything new from it because it's got all the information from the 747. Well, that's simply not true. I'm sure all of you know that if you want to develop an improved 747, say better streamlined, less air resistance, what you do is to use a wind tunnel model of a 747 to try to improve it. Now, that is use of model to predict a better 747. And if you want to teach a pilot how to fly, what do you have? You have a model of a cockpit, a simulation, and you teach the pilot how to fly using that model. Those are perfectly valid models, and they will give new information. So it's simply not true to say that models cannot produce any new information. They certainly can, because there are circumstances where we simply can't use a 747 to train pilots. At least they might well crash them. So I don't think Ray has produced anything in his talk that I can respond to in particular, and I don't think he's produced anything new. So I'll stop at that.
All right, let's see if I can get the computer to work.